Hello, everybody. My name is Arjun Deb. I'm a professor of medicine and molecular cell and developmental biology at UCLA. I also direct the UCLA Cardiovascular Research team at the David Geffen School of Medicine. And today I'm going to talk about the therapeutic opportunities in the cardiovascular space, which are being pursued in my laboratory, and some of the recent exciting advances we've made in this regard in collaboration with the UCLA TDG group. So, um, my lab broadly studies the biology of wound healing and tissue repair in many organs, and particularly in the heart. As shown here, the various aspects of biology and therapeutics we study range from the extracellular matrix to molecular targets for augmenting wound healing. We study a lot of pathological mineralization of tissues and how we can treat them. And then a large part of my lab studies drug development for various fibrotic diseases. So today I'm going to focus on two important aspects of therapy in my therapeutic opportunities in my lab. One is regarding the treatment of ectopic calcification, and then you will see how this morphs into another interesting area of how you can augment wound healing of tissues after injury. So talking about the first area, ectopic calcification, what do we mean from ectopic calcification? Broadly, it's an incurable condition that refers to mineralization of soft tissues. Normally, soft tissues are pliable and calcification is restricted to the bone, but in pathological states, soft tissues calcify too, and that disrupts organ function. It can affect the heart valves, and most commonly blood vessels. They're very common in highly prevalent conditions such as diabetes, age with aging as well, and chronic kidney disease. And the reason why we worry so much about ectopic calcification is multiple clinical studies have suggested that ectopic calcification causes accelerated mortality in all these patients who are predisposed to it. And what is the magnitude of the problem is illustrated in this slide. You can see on the left panel, this is a heart valve, which is heavily calcified. You can see these nodules of almost like little rocks of calcium. And obviously this valve does not function very well. And the only way you can treat it is by surgical or percutaneous replacement. This is a very common condition, the middle panel seen in patients who have chronic kidney disease. And you can see the middle muscular wall of the artery literally broken apart by these chunks of calcification. And this causes an enormous increase in mortality in these patients. We also look at various genetic conditions, rare diseases which cause calcification. And one particular disease we are focused in is PXE, or Pseudoxanthoma elasticum. And you can see here how the Bruch's membrane behind the retina is all broken up and calcified, and this causes progressive loss of vision in many individuals. So our story regarding ectopic calcification started several years ago when we had a paper in Cell Stem Cell where we identified this target called ENPP1. It's an ectonucleotidase, as shown in the middle diagram here. It hydrolyzes extracellular ATP into AMP and pyrophosphate, which subsequently cause altered pyrophosphate-phosphate ratios in the extracellular fluid and causes calcification to occur. And we demonstrated in this paper that if you inhibit ENPP1, you stop this process and you essentially prevent calcification from occurring. So based on the identification of a target, ENPP1, over the last few years, we've done a lot of small molecule development and now have identified leads which potently inhibit this molecule. In parallel, in collaboration with CROs, we have developed a monoclonal antibody specifically targeted to the catalytic site of ENPP1, and now we have humanized it, and currently it's in cGMP production. And so what this have resulted, it's sort of by attacking the problem with two different therapeutic targets. And here you can see in a mouse model of PXE how calcification is prevented in the heart by the administration of these inhibitors of ENPP1, which we have identified. And so this established proof of efficacy in preclinical models, and we've been fortunate to raise almost $5 million of direct funds from the federal government for IND filing of these therapeutic products. Moving to the clinic requires more than just a lab, and so we've understood that early on. And in collaboration with my colleague, Dr. Michael Jung, we formed a company, Westwood Therapeutics, along with Quang Pham and Jeff Cole, to further translate this technology to the clinic. And this was illustrated by the Innovation magazine as well. Now, what we are particularly excited is that lately, this ectonucleotidase, ENPP1, and this is from a recently published paper by another group, which shows that the ENPP1 is probably playing causal or pathological roles in many disorders. We've talked about calcification, but also the cardiovascular system, pancreatitis, diabetes, osteoarthritis, and so on. And so this led us to think that whether we can expand our therapeutic indications for ENPP1. And this is some really exciting data to show that ENPP1 can be used to prevent the development of heart failure, where we, in a preclinical model of heart failure, we administer the ENPP1 inhibitor for 14 days after inducing cardiac injury. And you can see here in the vehicle-treated animals, the ventricle heart is very dilated, and it's rescued when we administer the inhibitor, and is shown here in the echocardiogram as well. And this is just a quantitative assessment. You can see the animals which received the inhibitor in red have far better contractile function compared to those who did not. And before injury, of course, there is no difference in function. So we are excited about this, and we've moved on to really determine whether ENPP1 inhibitors can generally be used to prevent the development of heart failure and other aspects of wound healing. We've been fortunate to receive funding from the NIH, the NIAMS, and the Department of Defense to the order of almost $8.5 million. And we're now interested in looking at specific applications, example in diabetes, chronic kidney disease, PXC, and we're looking for phase 1 trial investment as well. What are the other things which are being developed in my lab? In the therapeutic pipeline, we have a monoclonal antibody which specifically reacts against immune cells to augment wound healing in the heart after heart injury. And we hope to propose a grant for that to the TDG in collaboration with the UCLA Metabolic Center. We have identified biomarkers which could be predictive of wound healing and how the heart will respond after cardiac injury, which can be used by clinicians to monitor adverse patients with an adverse outcome. And finally, we have expanded our role to the treatment of genetic diseases which cause dysregulated wound healing. And you can see in this mouse model compared to this wound is huge. And when we administer this drug, which we have identified in our laboratory, there is significant reduction in the amount of open wound. So these are some of the interesting things which are going on in my laboratory. I thank the TDG for this opportunity. And if you have questions, please feel to reach out to me. Thank you.